Hello everyone. This is a short video lecture about different models for DNA replication and the experimental evidences obtained from the famous Meselson Stahl experiment. At the end of this video, you should have a clear understanding about the different proposed models, a historical perspective of the experiment, methodology used in the experiment, the expected and actual experimental data obtained, their interpretation, and the different other conformational studies done to affirm the inferences from Meselson and Stahl experiment. First, we will see what were the different models for DNA replication that was proposed. The first model is called the conservative mode of replication. The other is semi-conservative mode of replication. And the third one is the dispersive model for replication. We will see first the conservative mode of replication. According to the conservative mode of replication, after replication there is 100% conservation of DNA between the daughter duplexes. It means that after replication, one of the daughter duplex will contain 100% parental DNA, whereas the other daughter duplex will contain 100% nascent DNA. So that is the nascent DNA. That is the proposal for the conservative mode of replication. Whereas in semi-conservative mode of replication, it says that there is only 50% conservation of the DNA between daughter duplexes. It means that after replication, each daughter duplex will contain both parental as well as the nascent DNA. That means one of the strand will be parental, and the other strand will be nascent. So but you can see, as from the figure itself, you can actually see is that each strand is either parental or nascent. There is no intermixing of the parental or the nascent strand. When you come to dispersive model of replication, what does dispersive model of replication says? It says that during replication there can be random double-stranded breaks in the DNA, and these double-stranded fragments will spin and rejoin in such a way that each strand will contain both the parental as well as the nascent DNA. So when you look at this duplexes, you find that there is a random inheritance of both the parental and the nascent DNA. So these are the three models for DNA replication. Conservative models says that there is 100% conservation of the nascent and parental DNA between the daughter duplex. Semi-conservative says that there is only 50% conservation. That means in each daughter duplex, one strand will be parental and the other will be nascent. Whereas in dispersive model, it says that it is random inheritance. That means each strand will contain both parental as well as the nascent DNA. Before we move into the actual Meselson and Stahl experiment, let's first dig into some history. The first person to do any sort of an experiment relating to the models for DNA replication was by Herbert Taylor. He done the experiment on an English broad bean. Basically, for the experiment, he used tritium labeled thymidine to radio-label chromosomes of the English broad bean. Then he followed the radioactivity in the succeeding generations in a non-radioactive medium. What he found out was that the radioactive containing entity in the chromosomes was maintained as a constant one from one generation to the next generation. He proposed that a chromosome may contain two such entities, because at that time it was not known that the DNA was a double helix or a quadruplex one. So he proposed that a chromosome may contain two such entities, and after replication each daughter chromosome is going to receive an original entity from the parental and a new entity. They use the word entity. Then came the Meselson's work. So this actually demonstrated that replication in eukaryotic chromosome is basically a semi-conservative one. It was earlier demonstrated. This experiment was done in 1953. In the same year, Watson and Crick proposed their double helix model for DNA. 5 years later, in 1958, was the famous Meselson and Stahl experiment conducted. The famous scientific historian Lawrence Holmes called the Meselson and Stahl experiment the most beautiful experiment in biology. Meselson and Stahl basically work for different laboratories on different topics. Meselson was actually interested in the sedimentation of macromolecules based on their density, whereas Stahl was interested in bacteriophage genetics. Both of them met at a summer training camp under a tree. This photograph actually shows the same tree they first met. This photograph but is actually taken 42 years after they first met. For the experiment, what they actually planned was to use a molecule called as 5-bromouracil as a density label to mark the DNA. But the problem with bromouracil is that bromouracil is a thymine analog. It is basically a thymine analog. It is a mutagen also. They basically cause transition mutations in the DNA. So due to the mutagenicity of 5-bromouracil and the cellular toxicity they can create, they opted for another kind of label, which was actually nitrogen. What they done is that they basically decided to grow E. coli in a medium that contains N15 as the sole nitrogen source. That was the basic plan of the experiment. So first we will see what is the basic technique used in Meselson and Stahl experiment. It is called an isopycnic separation. Isopycnic separation involves separation of molecules based on their equilibrating density with a column. The column used will be a density gradient column. It means that the stationary phase or the column used will have different layers that correspond to different density. If you add a molecule to such a density gradient column, what happens is that, and centrifuge, what happens is that the molecules are going to sediment or settle until their density become equilibrated or becoming equal to the density of the column. That was the basic principle of isopycnic separation. Two columns can be created. One is a sucrose gradient column. A sucrose gradient column is created by using different concentrations of sucrose. Percentage levels in sucrose can create the gradient of sucrose. The another is cesium chloride gradient column. In Meselson and Stahl experiment, cesium chloride gradient column was used because it is more convenient and easy to prepare. Why? Because cesium chloride is basically a salt. They will ionize in solution as cesium plus and chloride ions. Cesium have very high atomic mass, it is 132 atomic mass unit, whereas chloride have relatively lower mass, which is 35 unit. So if you add cesium chloride to such a centrifuge tube and centrifuge, the chloride ions will be uniformly distributed, homogeneously distributed throughout the tube, because chloride ions are lesser influenced by gravity. But when you look cesium, because they have higher atomic mass unit, as the centrifugation is going to proceed, more cesium ions are going to settle on the bottom of the tube, and as you move towards the top, the concentration of cesium is basically going to decrease. So upon centrifugation, you automatically create a density gradient column based on the concentration of cesium ions, so that the bottommost part of the centrifuge tube will be having higher density, because of the presence of more cesium ions, whereas as you move towards the top, the density is going to gradually decrease, and at the topmost you have a lesser density. So this is how you are going to create a density gradient column. This is the exact way how Meselson and Stahl created a cesium chloride density gradient column. In the Meselson and Stahl experiment, what they have done is that they have added the DNA molecules under study to such a cesium chloride solution, and they will centrifuge it. The cesium concentration is automatically going to establish a density gradient system inside the tube, so that the added DNA molecules are going to settle according to their density with the column. When the density of the DNA is going to become equal to the density in the column, they will stop settling and remain there. Then this tube will be shown to UV light at 260 nanometer. The wavelength that was used was 260 nanometer, because the lambda max, the maximum absorbance shown by nucleic acid peaks at 260 nanometer. So this particular test tube will be shown to a UV light. UV light naturally has a purple color, whereas where in the tube where the nucleic acid is located, the nucleic acid will absorb that UV light. So only in the location where the DNA is located, you will be finding a dark band, whereas all other regions will be purple blue color. Okay, so this the meaning is you are going to add DNA sample onto this test tube, shown it to a UV light at 260 nanometer. All the regions of the tube will be purple color except the region where the DNA is present, because the DNA is going to absorb that UV light and they appear as a dark band. Now let's move into the actual experiment. Consider this as our stock culture. It contains E. coli cells that were grown in a synthetic medium containing ammonium chloride, in which the nitrogen is the heavier isotope, which is N50. The natural isotope of nitrogen is the lighter one, which is N40. The bacteria were grown in this particular media for several generations, that all the biomolecules that contain nitrogen will incorporate N15 as their nitrogen, like the DNA and the proteins. Then these cultures, when transferred into a medium that contains the lighter isotope of nitrogen, which is N40, at different times, say I am writing some arbitrary time intervals. The first time interval is t is equal to 20 minutes. then the T is equal to 40 minutes, 60 minutes, and 80 minutes. So at different time intervals, DNA were extracted. DNA were extracted from these cultures and isopycnic separation was done as described previously. So from all these cultures, DNA were extracted and isopycnic separation was done. Now let's move into the analysis of experimental data. First, we will see what will be the expected results for isopycnic separation for the three models that were stated earlier. The extreme left centrifuge tube is actually contains the parental DNA. It's basically have a parental DNA. Consider this pink color as a DNA that contains N15. So you can see a single parental duplex that is in the stock culture, and they all incorporated N15 into their nitrogenous bases. So the duplex that you get in the parental DNA is simply called a heavy DNA. Simply because all the bases in this duplex will incorporate N15. So you start from the parental culture. In the parental culture you have only a single duplex, and in that single duplex in both strands all the bases will incorporate N15. So naturally the density of this DNA will be higher, so you are going to classify it as a heavy DNA. If you do isopycnic separation in the cesium chloride density gradient column, you would expect the DNA to settle or sediment onto the bottom of the tube. Why? Because the DNA is actually much denser. So I'll simply represent a heavy band at the bottom of the tube as in this way. So say this is our, simply imagine that this is our, so I'm simply saying that this is our heavy band. This is our heavy band that corresponds to the heavy DNA. So I think you have understood why you got a single band at the bottom of the tube, because the DNA was denser. So the top of the tube was actually lighter. So the DNA added onto the top of the tube will gradually settle until they are going to sediment onto a layer where their density is equal to the density of the column. Now let's first analyze if the mode of DNA replication was conservative. What will be the expected banding pattern in several generations? So we already know that only the parental generation was grown in the N15 medium, then all the other generations were grown in N14. So here represented are the samples collected from first generation, meaning the sample you have extracted from after 20 minutes. The second generation, meaning 40 minutes, third generation 60, and fourth 80 minutes. So these are the different tubes that corresponds to the DNA extracted at different times during our experiment. Now, what will be the number of dotted duplexes formed after each round of replication? So you can use this formula. The number of duplex DNA produced after replication is simply equal to 2 raised to n, where n stands for the number of generations. So if you look at the first generation, the generation is first one. So the total number of DNA duplex that is formed will be 2 raised to 1, which is 2 duplexes here. So you know that in the first generation replication is actually happening, the E. coli cells are in a medium that contains N14. So all the newly formed DNA will incorporate the lighter isotope. According to the conservative mode of replication, there must be 100% conservation in the inheritance of these nitrogens. Accordingly, you find that there are two daughter duplexes. One of the daughter duplex still contains only N15 and the other daughter duplex contains N14 only. Imagine that the blue color represents N14. So you can actually see that there is 100% conservation. Only one of the daughter duplex contains only N15, whereas the other duplex contains only N14. Since both the strands of this daughter duplex contained N15, still their density is heavy, so you represent it as a heavy DNA. Whereas both the strands of this daughter duplex inherit N14, the nascent DNA, so they are basically lighter than the parental duplex. So you should actually have two types of duplexes with two different density. One is heavy as the parental and one is lighter compared to the parental. So how many bands do you expect for the first generation? Is you expect two bands. One band is at the bottom of the tube that corresponds to the heavy DNA, and you expect another band at the top of the tube that corresponds to the light DNA. Their thickness will be the same. Since the concentration of both the duplexes are the same. So you will have a heavy band that corresponds to the heavy duplex and a light band that corresponds to the light DNA. Coming to the second generation, what you find is that it is these two daughter duplex that replicate into the second generation. So what you find is that when this heavy DNA replicate, you will have two daughter duplexes, and one will be the heavy one and the other will be the light, because the new nascent DNA always incorporates the lighter isotope. Whereas the daughter duplex in the first generation, which already has N14, they actually give rise to two daughter duplexes which already have N14. So this will be light and this also will be light. So how many bands do you get? Like the first generation, we get only two types of bands. One corresponds to the heavy band at the bottom, with the same thickness as the first generation, because the concentration is the same. Whereas when you go to the light band, what you find is that the thickness of the light band will actually increase, and the thickness of the light band will increase because the number of lighter DNA duplex has increased. So what is the ratio? What is the expected ratio you find here is that you will get one heavy and three light bands, three light duplexes are obtained. So the heavy to light ratio for the second generation will be 1:3. Coming to the third generation, how many daughter duplex do you get? The third generation means it is 2:3. That means you should actually get 8 duplexes. And who are those duplexes that contribute to this? It is the 4 daughter duplexes in the second generation that replicate to form the 8 duplexes in the third generation. And we already know that from this heavy duplex, you are going to get one heavy and one light daughter duplexes. Whereas from all these three light duplexes, you are going to get 6 light duplexes. So totally what you are going to get will be 1 heavy is to 7 light. You can actually see when you compare to the second generation, you can find that the concentration or the proportion of the heavy band still remains the same, but the concentration of the light duplex has increased 7 times. So the banding pattern still remains the same. In the third generation, you are going to find a heavy band with the same thickness as the second and the first generation. Whereas when you go to the lighter band, it has become more thickened because the concentration has increased 7 times compared to the heavy DNA. So this will be the banding pattern you observe. Now coming to the fourth generation, you can use the same logic. You can see in the fourth generation there will be 2 raised to 4 duplexes, which is actually 16 duplexes. And you find that the concentration of the heavy band is still the same. So you are going to find one heavy and the rest 15 duplexes will be light. So you can actually see from the first generation we have started from 1:1. We have started from 1:1 ratio, then it changed to 1:3, then it changed to 1:7, and then finally changes to 1:15. So you can find in the fourth generation also the thickness of the heavy band is going to remain the same, whereas the thickness of the light band has still increased, because now there are 15 times more lighter DNA, 15 times more lighter duplexes than the heavy band. So you can actually conclude that if the replication is actually conservative, apart from the parental generations, all the generations will contain two bands. One will be the heavy band and other will be the light band. As the generation increases, the concentration of the heavy band will remain the same, whereas the thickness of the light band keeps on increasing. Now let's check semi-conservative mode of replication. We have already said that in semi-conservative mode of replications, the parental duplex separate act as a template for the synthesis of the nascent DNA, so that there is 50% inheritance of the parental DNA between the daughter duplexes, as we have already explained. So in the first generation, what you get here will be 2 daughter duplex, and each daughter duplex will contain the parental DNA, which is actually N15, as well as the nascent DNA, which is N14. So when you look at the density of this DNA, it is going to be lighter than the parental strand, which is a heavy one, whereas it is going to be denser than the light DNA, which contains both the strands with N14. So you will have this DNA with an intermediate density. You will call this as a hybrid DNA. So in the first generation, what you find is that you are going to get 2 daughter duplexes with the intermediate density called as the hybrid DNA. So where do you expect in the isopycnic separation, where do you expect the band? We have already seen in the conservative mode that one will be at the bottom, that corresponds to the heavy band, and one will be at the top, that corresponds to the lighter one. But here you are going to get a hybrid band. You are going to get a hybrid band. I am simply writing it as a Y band, which is the hybrid band. This will be your expected observation for the first. Now coming to the second generation, you can find that there will be 2:2, there will be 4 daughter duplexes will form. So from this first generation, how many daughter duplexes will form? If you see, you can find that if you consider this daughter duplex alone, its heavy strand can actually give rise to a hybrid one, because the opposite coming complementary strand will be of the lighter N14, whereas the light strand of this duplex will incorporate another light strand during the next round of replication. So this single daughter duplex will give rise to another hybrid DNA and a light DNA in the second generation. Similarly, this hybrid DNA will give rise to 2 such daughter duplexes. One is a hybrid one and other is a light DNA. So what is the expect? So you are going to find 2 types of DNA here. One is the hybrid density DNA and other is the light DNA. What is the proportion? You can see it is 50-50. So you can see you are going to find 2 hybrid bands and 2 light bands. So you find the ratio as 1:1. right, two hybrid bands and two light bands, which is in the proportion as 1:1. So when you move on to the second generation, what you find will be, you are going to find a light band. You are going to find a light band. A new band will appear in the second generation, and similarly you are going to find a hybrid band with the same thickness as the first generation, because you can see in the first generation you had two hybrid, in the second generation also you have two hybrid. So you will have a hybrid band as well as a light band with the same, both will have the same thickness. Now coming to the third generation, you have 2 raised to 3 8 daughter duplexes will be obtained from the daughter duplexes of the second generation. You can find that it is now easy to calculate from the two hybrid duplexes in the second generation. How many daughter duplexes can you get? 4. And what are the 4 daughter duplexes? We have already seen from the first to second generations that 2 hybrid DNA can result in 2 hybrid and 2 light. So you can actually find that definitely you are going to get 2 hybrid and 2 light from the hybrid duplex, as well as from the lighter duplexes you are going to get 4 light DNA. So totally 8 duplex. Out of that 6 will be light duplex and 2 will be the hybrid one. So so you have 2 hybrid and 8 light, sorry, not 8, it will be 6 light DNA duplexes. So from the ratio in the second generation, which was 1:1, now the ratio is changed to 1:3. So you find that the banding pattern is still the same. You are going to find 2 bands. One is the hybrid band and other is the light band, but the difference is the thickness of the light band is now increased because there are 3 times more lighter DNA than the hybrid one. Now coming to the fourth generation, similarly you are going to get 16 duplexes here, but you can simply calculate, you can find out that there will be no differences in the number of hybrid duplexes formed. Still there will be 2 hybrid duplexes formed, and the rest 14 will be light DNA duplexes. And so you can see that now the proportion is still more separate. You can see now there is 1 hybrid duplex is to 7 lighter DNA. So still now you have 2 bands. One is the hybrid band with the same thickness as the first, second and third, but now the thickness of the light band has now increased because there are 7 times more lighter DNA than the hybrid one. So this is the banding pattern that you are going to observe in the semi-conservative. So what's the characteristic? The characteristic is that in the first generation you are going to find, in the first generation you will find that the heavy band that was observed in the parental DNA is disappearing and a new band is going to appear, which is a hybrid density band. And the second generation, a second band is going to appear, that will be the light band. Thereafter the banding pattern is the same, but the thickness of the light band keeps on increasing from one generation to the next generation. Finally, coming to the dispersive mode of replication, we have said that in dispersive mode of replication there are random double-stranded nicks in the DNA that allow their rotation and resealing, so that every strand of the DNA will contain both the parental as well as the nascent DNA. There is random inheritance of the parental DNA. That is the dispersive model. So what you find here will be from the parental DNA, after the first generation you are going to find 2 daughter duplexes, and when you look at the daughter duplex, what you can find is that each strand contains both the pink as well as the blue, indicating that each strand contains N15 as well as N14. So what will be the density of this daughter duplexes will be, they will be lighter than the parental DNA because they contains more N14 than the parental, but they are more denser than the light DNA because they contain some of the parental N15 also. So like a semi-conservative mode of replication, you are going to find a new band appearing in the first generation, and that will be a hybrid band. Now coming to the second generation, you find that there will be 4 daughter duplexes formed. If the mode of replication is dispersive, still there will be random inheritance of the parental DNA. But because 2 generations has passed by, and for the 2 generations this E. coli is in a medium that contains N14, what happens is that when you look at each strand, there will be more N14 bases incorporated relative to the first generation. Why? For the first generation, the bacteria has remained in that particular media for say 20 minutes only. But when you come to the second generation, they have been there for more than 40 minutes. So meaning that more N14 will be incorporated into these DNA. So what you find here will be that 4 daughter duplexes will still contain the N15 from the parent as well as the N14 from the nascent, but the amount of nascent DNA definitely would have increased for the second generation. So what you are going to get will be still a hybrid band, but the density of the hybrid band is not the same as the first generation because they have incorporated more N14. So you can find that. So this should be the actual position of the hybrid band if the density was the same as the first, but you can see that now the hybrid band is still shifted up, meaning it has become more lighter. This is your observation for the second. Now coming to the third generation, you find that there will be 8 daughter duplexes, and all of these 8 daughter duplexes will be hybrid. Why? Because they still retain the N15 randomly among them from the parent. But what happens is that all these 8 daughter duplexes will now contain more N14 incorporated than the second generation, but their density is still a hybrid one. So you find still a hybrid band whose thickness has now increased because in the second generation you had only 4 daughter duplexes. Now you have 8 daughter duplexes, so definitely their density, their thickness have increased, but their density has slightly decreased. Why? Because from the second to third generation there were more N14 incorporated. So you can actually find that the band has now become more lighter, and the band has become now more lighter than the second generation. Now coming to the fourth generation, you will have 16 duplexes, and if you follow the similar logic, you find that all the daughter duplexes will be still hybrid, but they have more N14 now than the third generation. So you find that the band thickness has now increased very much because there are now 16 daughter duplexes in this sample, but it has become more lighter due to the incorporation of more N14, but still the thickness, still the density of the band is actually hybrid. They are more they are more denser than the typical light band, and they are uh, lighter than the typical heavy band. So when you look at the dispersive mode of replication, you find that in all the generations, including the parental generations, you will find only one band. In the parental generation, that one band is the heavy band, whereas when you come to the daughter generations, first, second and third and fourth, there will be only single band that corresponds to the hybrid band. But the peculiarity is that the hybrid band is found to migrate from the bottom of the tube to the top as generations pass by. Why? Because as generations pass by, more N14 is getting incorporated, so the thickness of the band is increasing as well as the density is becoming more lighter. So this shows the overall data that is expected for each specific model for replication. So you can see that each banding pattern is characteristic for that particular model. But you can see a special characteristic when you look at the first generation. If you identify the semi-conservative as well as dispersive model for replication, you can see their banding pattern is the same. That is, for the dispersive as well as the semi-conservative, you get a single band that corresponds to a hybrid density. Whereas for conservative mode of replication, you should get a heavy band as well as a light band. So imagine if the Meselson and Stahl experiment were conducted only for a single generation. If the experiment was conducted for only a single generation, definitely you can discard conservative mode of replication. Why? But you cannot distinguish a semi-conservative from dispersive, because in the first round of replication, because we already know that now the replication is semi-conservative. But if the experiment was conducted only for the first generation, definitely you cannot distinguish a semi-conservative from the dispersive, because their banding pattern is the same. So we can state that in the first generation, the model you can discard is the conservative mode of replication, because you are never going to get two bands for the first generation in the actual experiment, because all DNA replications are semi-conservative. Whereas when you go to the second generations, you will be able to distinguish between a semi-conservative as well as a dispersive model, because in semi-conservative you will get two bands. One is the hybrid as well as the light, and we know for the dispersive, regardless of the generations, you will get a single band only on. So to confirm that DNA replication is semi-conservative, Meselson and Stahl experiment should be conducted at least for two generations. In the first generation, you can discard the conservative. In the second generation, you can discard the dispersive model for replication. Now we will turn our attention to the actual experimental data obtained in the Meselson and Stahl experiment. First, we will see the photographs of the DNA bands. We have already stated in isopycnic separation that the location of the DNA in the tube can be found out by dark bands. So when you look at this, this is the parental tube, and you can see there is only a single band, and that band corresponds to, you can actually see it is a heavy DNA. So this particular band in the parental generation, that corresponds to heavy DNA. So but when you come to the first generation, we have already said that what you should get is a hybrid band, and similarly you can see that there is the parental band is disappeared, whereas a new band is observed in the first generation, and it corresponds to what? A hybrid one, intermediate density N15 N14, and you can see that is the first one. And when you come to the second generation, you can find that another second band is appearing, which is corresponding to the lighter one. In the second generation, this is what you observe. When you come to the third generation, you can find that the hybrid band thickness is going to remain the same, whereas the thickness of the light band will keep on increasing, as we have already stated. Now coming to the optical density measurement, the OD values for these different DNAs. The OD value is directly related to what? The concentration of the DNA present. So in the parental generation, you can actually see an absorption peak that corresponds to the heavy DNA. When you come to the first generation, you can find another absorption peak, but that is actually shifted to an intermediate density. When you come to the third generation, you are going to find a 3 absorption peak at 2 absorption peak at 260 nanometer, one corresponding to a hybrid DNA and the other corresponding to a lighter DNA. When you come to the third generations, you can find that the OD value for the lighter DNA has increased, because there is more concentration of the light DNA present, and as the generations are going to increase, what happens to this peak? This peak is going to keep on increasing, because the concentration of the light DNA will increase from one generation to the other generation, whereas the peak that corresponds to the hybrid DNA remains the same. Finally, we arrive at the confirmation experiment, which confirmed the findings of the classical Meselson and Stahl experiment. This experiment was also conducted from Meselson and Stahl itself. What they did was they actually did a thermal denaturation experiment along with the classical principles of Meselson and Stahl experiment. So as we know, so what is different in this experiment is that we are actually, before doing a cesium chloride density gradient experiment, the DNA is subjected to 100 degrees Celsius temperature. So there will be denaturation and the two strands of the DNA are going to separate and then centrifuged. So this is the difference from the classical experiment. So here, before doing the isopycnic separation, first you are going to denature the DNA by heating it, so that the strands of the DNA are going to separate into two single strands, and then the isopycnic separation. What will be the observation for this experiment? If you look at the parental generation, which is the first tube, what happens if you do this experiment on the parental DNA? It is a heavy DNA. Both strands of the DNA are heavy. So they are going to separate it into two single strands, which are both heavy. So that if you do the isopycnic separation, still you are going to get a band which corresponds to the heavy strand DNA. Okay, that is what you are going to get. You are going to get a band that corresponds to the heavy strand. Whereas when you do that experiment in the first generation, what happens is that in the first generation all the daughter duplexes are hybrid. If you do that experiment, the hybrid duplexes are going to denature. It is going to denature, and what you are going to get will be one heavy strand and one light strand. That is what you are going to get, correct? So because all the hybrid duplexes will separate into single strands, so how many bands do you get? You get a band that corresponds to a heavy strand, as well as another band that corresponds to the light strand. Right. When you go to the second generation, still the same result. What you are going to get will be another heavy strand. I think you understood what is the logic behind this one. So the hybrid will separate it into a light strand as well as a heavy strand, whereas all light duplexes will give what? Light strands. So what happens is still you get a heavy strand band as well as a light strand band, but the thickness of the light strand will actually increase. When you go to the third generation, still the same result. What happens is that still you get a heavy strand band as well as the light strand band whose thickness is again increased. So I think you understood this data. So here you are not going to get any hybrid band because you are doing thermal denaturation before the isopycnic separation. The same result can be obtained. If you do the same experiment with the alkaline pH. Alkaline pH can also denature DNA. So instead of thermal denaturation, if you raise the pH to an alkaline pH, still the double duplex DNA will separate it into two single strands, and the results obtained will be exactly same. This proved that there is no intermixing of the strands in a duplex. Each strand corresponds to what? Either the light or the heavy strand. I hope the information provided here was useful to you. If you are interested in more videos, please consider subscribing. Thank you.